So in 1961, Mary Lyon described the concept of what's known as X chromosome inactivation, which is sometimes called Lyonization, after her. And what the Lyon hypothesis is that at an early stage of embryo development, in a female embryo, and this is limited to mammals, because remember, mammals have the XX, XY sex determination system. At a very early stage of the embryo, one of the two X chromosomes in a female embryo shrivels up and becomes inactive, meaning none of the genes on that X are going to be expressed, are going to be used. And the way we explain this is it's a mechanism for the cell and for the body to achieve what's called dosage compensation. You really need to think of the genes on the chromosomes as interacting the way ingredients interact in a recipe. If you're baking a cake, and I always like to use like a pound cake recipe as an example because I can remember the recipe. Because a pound cake recipe is 1 pound of butter, 1 pound of eggs, 1 pound of sugar, 1 pound of flour. You can probably put a little vanilla extract in there too, but it's a simple recipe, so I can actually remember it. So what if you think about genes that are on different chromosomes, not alleles of the same gene, but different genes on different chromosomes, or even on the same chromosome, different genes produce different gene products, proteins for the most part. And those gene products, though, interact together to create phenotypes. There's a lot of interaction between the gene products. And just like there's interaction between the ingredients in a recipe, it has to be at a certain ratio. It has to be at a certain molar amounts. So for a pound cake, for example, if you had a pound of sugar, a pound of flour, a pound of eggs, but only half a pound of butter, then something's going to be wrong with that pound cake, because the dosage, the amount of butter, isn't interacting correctly with the amounts of everything else. So if you think about it that way, females have 2 doses. We make twice as much gene product, in theory, because we have 2 X chromosomes. We would make twice as much gene product for the genes on the X chromosome as a male cell would. But yet, all the other chromosomes are in 2 copies between the male and the female. So what you would end up with is you would have to have a different way for the cell to function in a female body as it would in a male body. And things don't work that way. Most of your cellular functions are exactly the same, no matter if the cell is from a male or from a female. And so what happens, at least the way we understand this, is that by the cell inactivating one of the X's in a female cell, one of the X's shrivels up. That way, the recipe is the same for everybody. In other words, a female cell actually only has 1 active X and 2 active copies of every other chromosome, which matches what a male cell has. And so that way, it makes things equivalent so that the cell can, both a male and a female cell, can use the same recipes, so to speak. Now, the inactivated X chromosome in a female cell is called a Barr body, and that's a different scientist whose name is Barr. So the Lyon hypothesis is the whole concept of X chromosome inactivation. The Barr body is just the name given to the inactivated X chromosome, which you can typically see if you use the correct stain. You can see it inside the nuclear membrane of a cell. Now, this is a nucleus of a female cell, and they're giving you a little arrow to help you see that's a Barr body. In a cell that doesn't have a Barr body, this is the nucleus. I mean, you can see the nucleolus, but the Barr body would be sticking right against the inner side of the membrane, of the nuclear membrane. Now, when they do this staining on individuals who have certain numbers of sex chromosomes, they confirm that that X chromosome inactivation is a thing that the cell really does. And so, as you would expect, if you have an individual with 2 X chromosomes, they would have 1 Barr body per cell, per nucleus, actually. If you have, though, a Turner's female, she has no Barr bodies because her X is there's already just 1 per cell, so no need to inactivate anything. In the Klinefelter's male, you have 1 Barr body per cell. So normal male you don't, but in Klinefelter's male you do. And there's different forms of Klinefelters, so here's some other forms. But the rule is no matter how many X's you have, all but 1 will be inactivated in each cell. And we call that inactivated X chromosome of our body. Now, I stole this picture from somewhere online. It's pretty decent. It's pretty much what I would draw badly on the board if I were teaching you in a classroom. So, you remember when you were first conceived, one of your X's came from your mom, one of your X's came from your dad. Now, this is just for a female. Male zygotes don't go through this. But for a female zygote, as that cell divides into a 2-cell embryo and then a 4-cell embryo and then an 8-cell embryo, and actually it's not until about the 16-cell embryo, so here I know they only show 4 cells on this row, but pretend like this is 16 cells. The 16-cell embryo, at that moment, or at that time, in each cell there's a random decision, it appears to be random, in which one of the 2 X's becomes inactivated. It becomes a Barr body. So they're showing that with this little blob. And, statistics says that for any embryo, it's pretty likely that about half of the cells will inactivate the maternal X and about half of the cells would inactivate the paternal X. But, it's random, and so it's possible to get an embryo that has out of 16 cells, has 10 maternal X's inactivated and only 6 paternal X's. I mean, that's possible. But more or less, it's going to fall on a bell curve, and mostly you'll get about a 50-50 rate of inactivation between the maternal and the paternal X. But, I mean, if you really track it out, each of these 16 cells keeps dividing, and makes up certain tissues in the body. And so at the very bottom of this picture, in a really crude way, they're showing the final adult, or the final structure, the final individual. And so what you'll have is a patchwork of tissues, so to speak. Some tissues can track their history back to a cell that inactivated the maternal X. Other tissues can track back to a cell that inactivated the paternal X. I think I said that correctly. And by the way, when these cells, after the X chromosome is inactivated, every time these cells divide, they will remember which one was inactivated, and after a cell divides, it will then inactivate again each time. So what I'm saying is that from the 16-cell embryo forward, through every cell division forward, each lineage of cells will always remember and inactivate that same X chromosome forever. The only exception to this rule are the cells that make up the germline, the reproductive cells. They don't do the inactivation, but they're going to make the gametes. So, but for the somatic cells, you end up with an individual who has some tissues that can only express alleles from the paternal X and some tissues that can only express the allele from the maternal X. And so this gets you some interesting things going on. I asked the question, why are tortoiseshell and calico cats always female? Well, it turns out that it's an X-linked gene. The color of the fur, the orange and black color, is a single gene with two alleles. It may be more than two, but two alleles that are interesting here. So the gene has two alleles, a black allele and an orange allele. And you can't tell which one's dominant because you can never have a cell that has both alleles active at the same time. So if you have a heterozygous individual, which has to be a female because it's two X's, then one of the X chromosomes will inactivate in some of the cells, the other X chromosome will inactivate in some of the other cells, and each patch of skin will only be able to express either orange and other patches of, or black. Other patches will express the black. And so you can't have a heterozygous cat must be female. If you had a male cat, he would only have one egg, so he would either have the orange allele and he'd be solid orange, or he'd have the black allele and he'd be solid black. But a female can have both alleles because she has two X chromosomes.